Buddy, can I pour you a tea? Some coffee?
No go?
Yeah. I think I got to reboot this thing. It's something really... My keyboard somehow totally stopped functioning. You know, the cursor functions, but the keyboard no longer functions, and that's... But it was working, so somehow I'm plugging into the system here. It did some weird stuff.
Should I get you a tea or a water?
Tea would be great. Thank you. Thank you.
Hold that cup for you.
I've got enough I didn't spend the rest of the week, so I was sent it through the foreign jet.
You may not want a cookie, but there's a cookie anyway.
Looks good.
Oh, you're here too.
Dry, we'll just make $5 to part. Is it? Because I just didn't. I usually do that as I'm coming and I organise it with the reception. I'm such a lazy so. But yes, we did. It's been lovely. Yeah, gorgeous.
Okay,
Winter's
good.
day.
See, this is a new one. Never tell how these systems interact.
Did you do it or if you want to? Okay.
I've got my dog.
Yeah, why not? I think we'll need a minute or two for this, so we might as well let's kill it. So yes, go ahead.
He knows going somewhere. Clinging with you.
So it's easy as suitcases are going like.
That's right.
Yeah.
That's enough.
The classic.
I'm sorry, I only jumped in at the last.
This is the how to resell your Florida timeshare lecture, right? Yeah. Okay, good. Just want to make sure.
Yeah, I think it's okay.
Ta-da.
Yeah. It's an interesting paper, but I don't think people want to read that one. I've never seen it come up like that. It's up here. That's good. That's good. But let me see. Yeah.
That's up there.
Okay, good. We're okay. We're okay. We're good.
Welcome to this special ABI seminar, and I'm going to introduce the introducer. So Chris Moji, who many people here won't know, he's fairly new to New Zealand, has just become the person that heads the accelerator services part of Callaghan Innovation. And Chris is very involved in helping the development of medical device technologies in New Zealand. And one of the things he's done was to bring a number of people down here from the US as part of the workshop that we had yesterday, investment workshop. And one of the people that Chris has brought down is Buddy Ratner. But Chris, I'll leave it to you to introduce Buddy.
So I'm a biomedical engineer from way back, and I was also an entrepreneur and a venture capitalist, and I was living in Seattle, and of course of getting to know the community in the biomedical engineering department at the University of Washington. I had great fortune to become friends with Dr. Ratner and his wife Cheryl, and over the years we've started a number of companies together, and spent a lot of time kind of blue skying interesting ideas, and when this opportunity came up to bring my favorite American bioengineering researcher to New Zealand, there's only one guy I could think of, and it's a great privilege to be able to share his time, I think, with us here, so please give him a warm welcome.
Okay, and thanks, Chris, and return the compliment, and I'll tell you a bit more about Chris's important role in my thinking about entrepreneurship and commercialization. But mostly I want to talk about science, and I want to talk about medical devices and biomaterials. Is where this talk is really going to go. And but I thought I'd just introduce Seattle, because I'm very proud. I've been there 42 years now. Came as a postdoc, took one look at the city and said, this is not bad, I'm going to stay here. So Seattle is, let's see, this is going to work. So, yes, Seattle is there in the United States, called the upper left-hand corner. And it's kind of known for Boeing aircraft. Microsoft started in our area. Amazon started by us. Starbucks started by us. We're doing pretty well here. I don't know if you have Costco here, but they started that too. But also we're known for biomaterials. And again, that's kind of what I'm going to talk about. But this is just to give you some orientation. This is a picture from the air. This is called Puget Sound. It eventually would lead into the Pacific Ocean. So it's kind of a direct pathway to New Zealand. Some detours. This is our downtown area. The university is right over here. You know, without traffic, 5-10 minutes. And I live right there, right above this lake called Lake Union. And this is, you know, some of our views. We have Mount Rainier is a very prominent feature in our, in fact, this is taken from the deck in my house, so I kind of like that view, downtown. And this is our campus. We have also I think a very charming campus. The Japanese government, some 60, 70 years ago, donated cherry trees. We have a very beautiful quadrangle of cherry trees. So that's a bit about Seattle. I just thought I'd do some little show and tell there. But what I really want to talk about is these things called biomaterials. And biomaterials aren't very much used by themselves. We make them into medical devices. And this just illustrates a few of them. I'll talk more about other types of medical devices as we go along here. But just so everybody knows what I mean by medical device, particularly I'm interested in implantable medical devices. I should probably qualify that. And so let's see, medical devices and the biomaterials which they're made, it's projected to become a $434 billion industry. It's pretty big. But also, importantly, it will impact millions of patients, and is at the moment doing that too. And so these are some pictures of biomaterials. They're about as boring as could be. You know, it's kind of titanium, Teflon, silicone. These are widely used in many, many medical devices. But the word that comes to mind when looking at these materials is inert. And we're going to address this in a very interesting way. I mean, this is kind of what most classical medical devices are. We're going to ask a question. Can an inert polymer behave like a pharmaceutical or an active biological signaling agent? And so the roots of this talk do lie in biomaterials. So I'm going to go back in history, do some roots and such. Let's see, we have kind of an outline, a bit of history, talk about some issues and opportunities, new developments. But I like to think about how this subject so freely integrates some biology, some material science, medicine, and engineering. They all kind of come together in this subject. Okay, so let's start historically. And ancient Egypt is the place to start. So this is in the British Museum. This wooden toe is, I guess at the moment, stored someplace. But it was found in a mummy. So, you know, 3,000 years old. 3,000 years ago, people were thinking about how we'd replace a part. Big toe is actually very important for walking and for proper locomotion, proper gait. And more than likely, this toe implantation killed the person who it was intended for. Wood's going to result in a pretty intense reaction. But maybe they tried this experiment. The Egyptians also used linen sutures, probably some of the earliest instances of sutures, and also gold was used implants. So there's it's a very early history there, but of course there was no science behind that history. But this is another interesting footnote in history. It's a book that was published in 1938 called The Culture of Organs. And the authors are Alexis Carrel, is a French surgeon who won the Nobel Prize in Medicine, maybe 1925, I'm going to guess, roughly that time, working with Charles Lindbergh, the American aviator, who first person to fly solo across the Atlantic Ocean. But Lindbergh was an engineer. And they wrote this amazing book, which I think is maybe the historical precedent for tissue engineering and such. And this idea that we could develop bioengineering systems to keep organs and tissues alive. A very, very intriguing volume here. About the same era, this man in the center, his name is Wilhelm Kolff, and Kolff was an MD in Nazi-occupied Holland at the time. And Kolff, nephrologist, was thinking, maybe innovating would be the right word, about a historical situation. If somebody had kidney failure in that era, you died in about 3 weeks. It was not considered a pleasant death at all. And Kolff said maybe we could do something like that. Maybe we could filter patients' blood using a dialysis membrane. And in fact, although this illustration was a few years later, this wasn't in at the time of the original development, he used sausage casing. He used a washing machine tub with a dialysate, and demonstrated you could keep humans alive with dialysis. And this was the first time in history anybody was wasn't cured, but sort of restored from this death. Now the interesting thing that happened, I don't know if I would call it interesting, Dr. Kolf had to interface this dialysis system with blood. And at that era, it involved some major surgery. And in fact, the patient had 6 surgical sites they found that could access the blood appropriately. And after that, the patient died. They could no longer get access to the patient's blood to install the dialysis equipment, which has led to some very other interesting stories. So now we're jumping way ahead to Seattle. This is a plaque that, let's see, on this site in 1962, the world's first artificial kidney center was opened. And the reason why Seattle had the first artificial kidney center, which actually was in the basement of this motel near the university, is where they had the first kidney center. But the reason why is because of a couple of, again, innovators and pioneers. This man, Belding Scrivener, was a nephrologist and envisioned a way to get continuous access to the blood of patients. This man in the center, his name is Wayne Quinton. Wayne Quinton was an engineer and figured out how to build what Belding Scrivener envisioned, which was again, a blood access system that allowed them to just hook on the artificial kidney. And this man, Les Babb, was a chemical engineer. And Les Babb built the apparatus that controlled the dialysate, controlled the fluid flows, and all such things. So this team came together in Seattle, and that has led to a successful kidney dialysis. And so if we look at kidney dialysis today, we have very efficient equipment. You can kind of see a bit of this blood access here. And hollow fiber dialyzers these days, and very nice control systems. But in this first decade of the 21st century, we spent, that's 2000 to 2010, the world spent $2 trillion on dialysis therapies. And there are greater than 4 million people worldwide on kidney dialysis. Probably a lot of people in developing countries that simply just die because they don't have access to this kind of high-tech equipment. But there are 4 million people that are benefiting from this equipment. But interestingly, in the US, 50% of the people on dialysis do not survive 5 years. We hear of many cases of people who live 20, 30, 40 years on dialysis, but 50% of the people don't even survive 5 years. So there's a lot of opportunity here to think about how we can use all these synthetics and do better for our patients. Here's another interesting incident. So World War II has just ended, and there were a bunch of new materials, new polymers, new metals that were considered war necessities, military necessities, and were not available to the general public. And suddenly the war's over, and suddenly they're here. And one of the famous incidents is what's called the nylon wars. The nylon stockings were apparently a large improvement over silk stockings of that era. And there were almost riots at department stores when they were first released. But nylon, Teflon, silicones, titanium, stainless steel, these are all materials that were not available during the whole war. And suddenly they're there. And suddenly physicians look at them and say, these are very, very novel materials. They have stability. They have inertness. They have great mechanical properties, or interesting mechanical properties. I have a patient that needs this or this or this. And this is really, this release of these materials is really the origin of modern biomaterials and medical devices, made it possible. So here's another World War II example. This man, Harold Ridley, was a ophthalmologic surgeon for the Royal Air Force. And Dr. Ridley, at the end of the war, got to examine the eyes of aviators. The war is over. And of course, the eye is sort of a transparent, clear window into the body. Looked inside and saw what I'm going to call an unintentional implantation of shards of plastic due to the dogfights, the machine gun battles over the European theater that shattered the windshields. Many of these pilots received just shards of plastic that was blasted into their eyes. Years later, after the war, he looked in the eyes, and there was the shards of plastic kind of sitting in an inert fashion. And at that time, the conventional wisdom, well, you got a splinter, you know, it gets red, it gets painful, it gets swollen, hurts. But here are shards of plastic that have been in these patients for years doing nothing. And you know, Ridley kind of scratched his head and said, maybe this is biocompatible. He didn't use those words, but his writing implies that this is what it was. And that windshield observation led to the first intraocular lenses. And eventually, the greatest source of blindness on this planet was essentially cured. And Dr. Ridley was knighted for his contributions to civilization. The intraocular lens, this shows what it looks like. It's in the capsular bag, the bag that holds the natural lens. These little springs are called haptics, and they kind of stabilize the lens in place. And let's see, this is me after I had one of these lenses implanted in my eye. And you can see the pupil is dilated, so the surgeon could work on it. My surgeon colleague put her initials over here so she got the correct eye when she put the lens in. And the lens has functioned quite well, although I'm having now, I shouldn't be complaining about my medical conditions to you, but I'm having now a condition called a secondary cataract. And it actually is considered a biocompatibility reaction to the plastic, triggering prolific cell regrowth, which is, I actually have cells regrowing over my lens. And what I'll need to do is go in for a laser procedure. They literally blast away that cell layer. And apparently it doesn't hurt, but I'd rather not do it anyway. But I guess I have to, because that lens is starting to fail on me. Here's another one. The hip joint, hip prosthesis, and just many, many different styles and models of this. Again, this kind of inert metal, the titaniums, the stainless steels, the cobalt, chrome, molybdenums that are used for these devices. They obviously take a person who is essentially unable to walk, and within 2-3 weeks, that person is out dancing again. It's really been quite remarkable. And these are going into, if you take hips and knees, and the knees are really quite similar in design, you take those two devices and they're going in pretty much 2 million people every year, doing some amazing things. Here's still another one, the sort of infamous breast implant. And there's interesting stories here too. You know, it's a very unassuming looking device, really a silicone bag. But developed in the early 60s by this MD. By 1980, there were 100,000 women having them. In 1992, the FDA declared a moratorium on the use of silicone breast implants. There was concerns that there might have been some autoimmune diseases triggered by these devices. By 1996, the FDA kind of sheepishly apologized and said there really wasn't evidence for this at all. And there was no evidence that there was anything particularly wrong with it. Then the numbers have steadily increased, and I always thought it's quite interesting. The majority are cosmetic rather than mastectomy reconstruction. So, oops, let's see, go back one more. So what we've seen here is this kind of evolution, this interesting evolution in biomaterials research. Really, all the early innovations, the breast implant, Charlie's hip joint, the intraocular lens, the kidney, these all came from what I call surgeon heroes. Surgeons who didn't have the institutional review board type system we have now, and just had patients who needed things and tried some very heroic things, some of which worked, some of which didn't, but they were trying to do things for their patients. In the 50s and 60s, the surgeon hero dominated the field. By the 70s and 80s, we had engineers moving in, and then maybe sometime around the 90s, another type of engineer really raised themselves in prominence, which is the bioengineer, which would also add to the engineering, biologists, and some material science elements. Okay, so that's kind of a little bit of the history, taking it up, and let's talk about some issues and opportunities. Again, here's another chart with a list of a whole bunch of medical devices. I'm not going to read them. It would be very tedious, but again, just dwell on the magnitude of the numbers. And these are worldwide numbers, and these are how many are going into humans every single year. So, you know, with numbers this big, what would you say? You'd say that if they caused some disastrous, you know, if they didn't work, if they led to disasters, there'd be almost an epidemic with this many people getting things. We don't see that epidemic. Mostly these devices work. We have genuinely millions of lives saved and the quality of life improved for millions more. It's a pretty nice record for medical devices. Millions of lives saved, the quality improved for millions more. But I'm going to ask, is there a dark side to the story? And so if one looks at how well the medical devices really work, one finds some great costs to the health system and also some great challenges or traumas to the patients. So these intraocular lenses, the IOLs, which again I have one, they have a 25 to 50% operation rate. I suddenly find myself in that category. And so again, it's expensive and it's actually rather traumatic to the patient, me, for example. Hip and knee joints were developed in the late 50s, early 60s to have maybe 10 to 15 year lifetime, and they still have about that same lifetime. There have been no really substantial increases in the lifetime of these devices. So if a young person needs it due to a sports injury or a couple of diseases, degenerative diseases, that young person you know, might be 20. By the time you're 35, you're going to need another one. And they might be able to put that in, but then by 50, you'll need still another one. And to put a third one in, usually there's not enough bone left to really allow that. So big problems with that. Vascular grafts, blood vessel replacements don't heal, don't re-endothelialize, prevents us from using small diameter blood vessel grafts that are really needed badly. Heart valves calcify and thrombose. Percutaneous devices through the skin don't seal to the skin. Bugs get in, bacteria get in, and cause a tremendous amount of problems. Electrodes get encapsulated, they cease to function. Catheters, thrombosis and infection, stents, clotting and closure, contact lenses, simple device, but anybody who's worn contact lenses know how uncomfortable they can be, how occasionally it leads to red eye and pain in the eye. Dental implants loosen. The list goes on and on. In fact, it goes on so much, the US FDA has what they call their adverse events reporting system. And a manufacturer, by law, if they receive word from the physicians that a medical device is not performing well, they have to, by law, fill out a form, report it to the FDA. And so this is all on the web, and if we go up to the most recent data I could find, in 2011 there were 870,000 complaints about the performance of our medical devices. That's a pretty substantial number, just the US, just the United States, so very large number. So this leads to the slide of opportunities. Where are there opportunities? We'll talk a good bit more about improved healing and reducing fibrosis that I consider a considerable problem in many devices. But regeneration, reconstruction, and I might even put things like tissue engineering here, huge opportunities to change the way we do medicine today. Kidney dialysis, as I've said, tremendous opportunities. Heart failure, the #1 reason we leave this earthly sphere is due to heart failure. More people in developed countries died due to this. Diabetes, again, rampant problems. There are opportunities to develop infection-free medical devices. Infection is still a huge problem. And ultimately, reduce costs. We call in the United States now accountable or affordable care. Okay, so let's go back to biocompatibility. Again, maybe this observation that Dr. Ridley had in the earliest days. And so if this thing here, a biomaterial, at pretty much time 0 when it's implanted in the body, in as short a time as we could measure, there's going to be a layer of proteins that absorb, coat the material. And so really biology never actually sees our clever biomaterials. They see a layer of proteins that come down. At sometime around 48 hours, these cells called macrophages come along. Macrophages are programmed by evolution to get rid of foreign objects in our body. They get rid of bugs, they get rid of splinters, they get rid of foreign objects. They eat things. They produce powerful proteases and oxidation elements, and they attack things. And although we've put great effort into our biocompatible biomaterials, the macrophage is much unimpressed. It is a foreign object and just wants to eat it. So the macrophage tries to engulf it, to phagocytose it, and finds that silicone rubber, titanium, stainless steel, these are simply indigestible. It does not have the mechanisms to do that. So it leads to this spreading, the cells spread out. Again, they try and engulf it. When they find themselves frustrated in their inability to, it leads to encapsulation. They bring in fibroblasts, make a collagenous bag that surrounds the implant. And this is the reaction we see in all biocompatible biomaterials. So this is a scanning electron micrograph. It's an implant in a rack for 6 weeks. And this Teflon device is a glucose sensor. And this is, in a scanning electron micrograph image, this is what loose connective tissue or normal connective tissue looks like. It's pretty open. Glucose could readily diffuse through there. Lots of blood vessels could reach the glucose sensor. But look what the body does. It puts this dense collagenous wall surrounding the Teflon device. And indeed, we find implanted glucose sensors fail due to this capsule because they no longer have an ability to read or monitor the glucose levels in the bloodstream due to this encapsulation. So what's wrong with this encapsulation? Walling off electrodes. Electrodes lose their electrical properties. Sensors, biosensors, drug delivery systems no longer deliver drugs reliably because you've got to go through that dense capsule wall. Breast implants have a classic problem with this foreign body capsule. Can lead to poor bone healing, can lead to poor pacemaker healing. Vascular grafts get a thick sheath around them that I believe inhibits the healing. Device-centered infection, we can't get the blood cells in there to properly address the bacteria. So I think maybe infection can be associated with this too. A lot of problems with this encapsulation. And you find that this is an article in Expert Review Ophthalmology talking about glaucoma drains. And I'm not going to read the whole thing, but there's a commercial glaucoma drain that says reactive fibrosis results in the long-term failure of these devices. The clot, the scar, that foreign body capsule. Here's another example. We did some work with a company looking at continuous infusion of insulin or insulin pumps. That's actually not continuous infusion, but implantable insulin pumps. And we usually those go through the skin. They're percutaneous. But in this case, we implanted a Alzet mini pump directly in, let's see, I'm trying to remember if we did mice or rats, probably mice actually. And here's the catheter, the type of catheter that's used in humans. And what did we find? The company who we were working with wanted to know why their insulin pumps failed. Why could the patient only get five days out of their insulin delivery catheter? And the reason why is if you look at the foreign body reaction, you're starting to get blue in this. This is trichrome stain. Blue means collagen. We're starting to get dense collagen in here. If you look closely, massive macrophage attack. Classic foreign body reaction, probably exacerbated by the insulin coming out of those pumps. So they're just some examples and of the problems, but what this talk is not going to address is two approaches to get around this foreign body reaction. How can we develop a new generation of biomaterials that will not have those problems? And so one is going to be a novel, called an engineered porous scaffold, and the other will be a non-fouling surface. So let's see. Engineered porous scaffolds. So there have been observations in the literature going back to at least the 1970s showing that if you implant a porous material and the same material in solid form you'll get different kinds of healing for the two. You'll see very distinct differences. But there really wasn't a systematic set of rules about what's going on. So, and you know, this is a typical kind of porous scaffold, one that we made here. And what you examine this, you find that it has large pores, it has medium-sized pores, it has small pores. You know, typically people express the porosity of this as some average. But we asked, you know, if you really want to know what pore size was best, it'd be very hard to infer it working with materials like this. So I set one of my Ph.D. students, Andrew Marshall, on a quest to make a material that had one and only one pore size. And we developed this process which used microspheres that are sieved to get a very sharp size cut, a uniform size of microspheres. Then we ultrasonically shook them so they settled into a close-packed array. Then we gently heated them so the microspheres fused or sintered together. Then we surrounded them with a monomer solution and solidified that, and finally solubilized the microspheres out, leaving us with a material where every pore is the same size, where the spheres were sintered together. Now we had interconnects between the pores. And so we call these materials sieve, shake, sinter, surround, solidify, solubilize, success. And as we say, they have led to success. Actually, the student, Andrew Marshall, is a champion tennis player. And he's developed a 10-step process, which we call tennis balls now. But the puns go on. It's terrible. But this is what the material looks like. It has a nice uniform look. These are what the microspheres actually look like. These black dots are where the spheres were sintered together. So these are the interconnects. And this shows a big piece of it. So you can get a feeling for how uniform the pore structure is across these materials. And Andrew implanted them. He made these materials and implanted them in mice for a month. And we got a result that we don't think has ever been seen before in the biomaterials, in the materials medicine literature. Is on both these plots, the y-axis is vascular density. And if the pore size was something around 35 microns, we got impressive vascularity. If the pore size, and it fell off very, very quickly with pore size. So you can see here we're plotted with error bars. It's a very sharp maximum. And importantly, someplace up around this maximum, we got this sort of vascularized reconstruction, where for smaller or larger pores, we got this scar, the foreign body capsule. This shows it again. We had one of the referees for our paper was rather fussy and said, well, you're reporting blood vessels, but are these functional blood vessels? They have blood flowing through them. So Lauren Madden, for her thesis, part of her work, she injected a biotinylated lectin, which bonds to endothelial lining of blood vessels. And so the only way it would ever get into our implant if there were functional blood vessels. And sure enough, again, at the 40 micron, we saw this very distinct maximum. Let's see, another of my students has been exploring these. Eric Sussman, he turned out some very nice results too. This shows some of his data on, this is the non-porous, the control material. And this is the material. It's a hydrogel, by the way. Hydroxyethyl methacrylate, the standard sort of soft contact lens material hydrogel. And again, trichrome stain, which stains collagen a dense blue. So you can see this really dense foreign body reaction here at 3 weeks. The 35 micron pores, the reaction is radically different. Obviously, we have a lot of cellularity going on in here. Essentially none of this very heavy capsulation. And then by the time he went up to 160 micron pores, great big pores, you see again a lot more collagen. You can see some of the foreign body capsule forming. So it was a really pretty dramatic difference with this 35 microns being this nice vascularized healing. We also have done a bunch of work in putting these directly into heart muscle. And there's really nothing in the body that wants a scar more than heart muscle. And so on top here, these yellow bars are the foreign body capsule thickness. And we see the solid and the 60 micron have a rather thick capsule. By the way, 140 microns, the regulatory agencies, the FDA and such, would say this is acceptable. This is what biocompatible looks like. But this is a classic foreign body capsule. The capsules were much thinner on the 30 and 20, but there's more to it than just being thinner. Again, we're using this trichrome stain, and dark blue is collagen. So you can see around the solid material, very dense, very dark blue, 60 microns, very dark blue. Look at how light and diffuse the trichrome is around the 30 micron, and then it starts getting a little darker again at the 20 micron. So again, something very special about this pore size. We wanted to compare it to more conventional porous materials, so we compared it to Gore-Tex, to a classic porous Teflon. And so let's see, the top two are expanded Teflon. We have a stain for endothelial cells, and it's the brown color, and then the DAPI just looks at nuclei, so we look at total cell density. And notice how at the interface, very heavy density, and this is primarily macrophages if you examine them closely. Take the 30 micron 6S material, and again, I'll show you the quantification of the endothelial cells in a second. But notice how there's really hardly any interface at all here, as opposed to this dense line of macrophages we see in Gore-Tex. And here's the quantification of the staining, that brown color, showing that the porous, the 35 micron material, or 30 micron in this case, had much more endothelial cells than the classic Teflon material. There's one more example of this. This is from my colleague, Dr. Briers, and his group is working with this too. And one of his postdocs actually did what we would call a rather poor preparation of the materials, but it actually made for a very interesting example in that if you're not careful, you can get a solid skin over the implant, which prevents cells from invading and prevents this reaction from happening. So there's one part of the implant that was put in that had the solid skin, and that's over here. And you can see this dense foreign body reaction. But over here was the good material with the good pore structure. It didn't have the skin. And look how there's almost no capsule. Very, very dramatic example of how profound this is. Well, we started working with our dermatology group, MDs, and they're very involved with percutaneous implants. They developed a model where they could put a little rod of material in the back. In fact, you can see them actually, photographs of it in the back of a mouse. And with this type of model, you could, the rod of material actually went through like this. So you can do histology and look at both the epidermal surface and the dermis in it. And the conclusion of the medical group was sphere templated polymers with 40 micron pores demonstrate an ability to recapitulate key elements, both the dermal and epidermal layers of the skin. Read on, basically they said the skin just heals and reconstructs right through it, which is something they'd never seen in 30-something years of research in this area. Also working with an orthopedic surgeon, Paul Manor, and Paul's bone model is rabbits, but he doesn't like just rabbits, he likes old rabbits, because old rabbits don't heal bone very well, just like old people don't heal bone very well. And so these are 18 to 24-month-old rabbits, and there's a description of his procedure, but basically he drilled a hole in the bone. I asked him how he does the implants. He said, I took the material with my thumb and I stuffed it in the hole. It's a really elegant surgical procedure. In any event, we got some very interesting data from these rabbit studies. So the control, which was just this divot drilled in the bone, clot formed in there, but nothing really happened. It was a total mess, actually. But in our 6S materials, the bone just invaded and started growing in. So we did some micro CT on this, and so we could look at what normal bone looks like, and we can actually watch the healing zone in this divot in the bone. And when we did the quantification of the data, there's a couple of experiments combined here, but basically empty is just the unfilled divot, and filled is with our 6S material inserted. We have contralateral implants here. So empty, very little bone growth, filled, very nice bone growth, empty, very little, filled, very nice bone growth, empty, very little. Filled, one of them didn't work out so well. But anyway, 4 of the 5 actually look very impressive in terms of the ability to grow bone. So this is from the bone standpoint, the bone or orthopedic biomaterials standpoint, this was actually very striking results. And that the material was, again, this 2-hydroxyethyl methacrylate. People would say that's not a bone healing material. We had no, we used no cytokines like BMP2. We didn't put in any stem cells. We didn't put any osteoblasts in. We had no hydroxyapatite or calcium phosphate. And the conventional wisdom in the bone field is you need great big pores. You need 200 micron pores, and we did it with 35 micron pores. So pretty much every conventional wisdom, I think we pretty much challenged here. Also, we did a bunch of work on implantation in the sclera of rabbits. This is actually with the same surgeon who did my intraocular lens, but he's a very good surgeon. But spite of my lens family. It's not her fault. Biomaterials' fault. Okay. So anyway, what we saw was beautiful integration. Again, no scar. Sclera is another tissue that is very prone to scarring. We saw this beautiful integration into the silicone rubber. That was silicone rubber. We also used hydrogel with the same size pores, these 40 micron pores. So we had a hydrophobic material, hydrophilic material. And again, this also shows the hydrogel. And you can see here's the sclera and the conjunctiva, and it's implanted basically in the wall of the eye, in the white of the eye. And we look at a higher magnification. You can see our 6S structure here. Look at a higher magnification. We see this beautiful ingrowth of tissue and reconstruction. It's nicely vascularized and nicely healed. Very impressive reconstruction there. Well, this was so impressive that, thanks in fact to my good colleague here, Chris Moji, we launched a company called Helionics. And Helionics was a company set up to manufacture that type of porous material, this 6S material, which they rebranded STAR. They call it sphere templated angiogenic regenerative material. And Helionics then started working with a veterinary device company. So dogs get glaucoma also, and apparently in dogs, in humans you get glaucoma, your intraocular pressure increases, but you don't feel anything. You just gradually go blind. In dogs, apparently it's quite painful to the dogs. So dog owners and dogs are very interested in doing something about it. So this company developed they always had a glaucoma implant for dogs. It was a veterinary surgical company. But then they started making them out of our material. They called it Clarify. And where their classic dog glaucoma drain lasted maybe 4 months, 5 months would be typical. Some of these have gone for more than 40 months. So I was sort of asking them, we'd love to do some histology and see how they're healing. And they say, well, these are people's pets. We have to wait till, can't just give you their pets to do histology on. Well, anyway, it certainly worked well for them. And it worked so well, in fact, that we spun off a company to make these for humans. And the company is called iStar Medical. And they're making this device called a Ceton. And received its CE mark and is going into humans now. Initial results look very nice. Another piece of work is done by the Helionics company. Andrew Marshall is now the chief scientist. They're no longer my student, but did some nice work here looking at a cuff around of the STAR or 6S material around catheters in pigs. And let's see, the two control materials were commercial catheters, commercial percutaneous catheters. And what he looked at was the, let's see, so the orange and the red are the control catheter materials. And we're looking at catheter survival as a function of time, and both of these fell off. Where the star materials both had actually considerably better results. And this is in a large moving animal, so it really is kind of a challenging situation for a percutaneous implant. Also working with a glucose sensor company called Perfusive Medical that wants to do a fully implantable oxygen sensor, and Kristen Helton was really the lead. Dr. Helton was the lead scientist on this piece of work. We had a pretty big team here. But they wanted to fully implant an oxygen sensor and do long-term monitoring of oxygen in the body. And the sensor had a porphyrin in it that fluoresces with oxygen. And this is our sensor, and this is a conventional oxygen sensor of the type they're using now. So obviously we have some big size advantages. And so we used our 6S material. This is a fluorescent image of the extensive blood vessel invasion to our material. But looking at this sort of macro view, look at how nicely the blood vessels are invading our material. And the important results, and we're in the process of publishing this now. It should be submitted to a journal already. But let's see. We have our 6S material. It's the blue curve. We have the solid hydrogel with the fluorescent porphyrin, red curve. And then we have a commercial needle sensor, which is the black curve. And at 0 time, the responsiveness of all of these is, what this is they change the oxygen level, and there's and then reintroduced oxygen, reoxygenation. And the fall off was actually relatively slow, but it's about the same for all the sensors. Now then we go to 170-day implantation, and we can't use the commercial needle sensor, just to lead to terrible infection at that level. So we just have the solid hydrogel and the blue material, but the solid hydrogel has, again, this lag time. With the blue, now it's healed in a vascular manner, has almost instantaneous response to the change in oxygen. And 170 days is a pretty long implantation time. So let's see, these 6S materials, they've shown good tissue integration, subcutaneous, intramuscular, sclera. We're working with a gynecological surgeon, rebuilding vaginal wall, percutaneous in the heart, in bone. I won't even talk about tumor models. We can use many different polymers. We're not stuck with one polymer. The only thing important is that pore size. So ask, why are we getting this universal healing? So one of the observations, this is a stain from mature macrophages, and we find in the 35 microns quite a heavy invasion of these mature macrophage cells. And through most of history of medicine, there was one cell called a macrophage. But probably in the last 10 years, people have started thinking about different shades, different flavors of macrophages. They call them polarizations. And people talk about an M1 and an M2 macrophage. So the M1 is killing the parasites, tissue destruction. The M2 is angiogenesis, tissue remodeling. So maybe we'll get in the M2 macrophage. So part of the hypothesis why we might get the M2 macrophage is in the non-porous material, we get this frustrated phagocytosis going on. In the large pores, there's plenty of room for that macrophage to spread out. And again, going to the spread configuration. The small pores are large enough for the macrophage to get in, but not large enough to really swell, spread. So what we might be doing is sort of mechanically tricking the macrophage down a different pathway. And so are there M2s in these small pores? Well, these are some implants in heart muscle. And let's see. So all the gray bars are various porous hemas with different pore sizes. The non-porous is black. And so we see at the 40 micron, there's the most M2. The statistics are a little bit rough and ready here, but it kind of looks that way. The M1, in any event, the attack phenotype in the solid material, we've got lots of them, much less M1s. for the porous materials. And then there's this interesting mixed phenotype, M1, M2, that many other people have noted. And we got lots more of those in the porous materials than we do in the solid materials. So that was kind of interesting. Eric Sussman continued exploring this. This is just a micrograph showing, again, our 6S material. And here's the surrounding tissue surrounding the implant. It's just a light micrograph. But then he did a stain for macrophages, found about the same number of macrophages outside as inside the material. That was kind of interesting. But then he did a stain for M2 and found the M2 predominated outside the material. And if he stains inside, he finds M2 on the inside, M1 on the inside. This was totally opposite of what we thought. The data is very good. I'm very confident in this data. We're still trying to figure out exactly what's going on here. But anyway, the M2s, we don't see this in a solid implant. We don't see these M2s outside. Somehow that porous implant is triggering this big population of M2s. So again, we don't know quite what's happening. And there's a couple of slides that kind of document with a variety of different stains for M1 and M2 that continue to support this idea of M2 on the outside, M1 on the inside. And I don't have time to show them. So I'm going to go through. And so there's real data backing this stuff up. It's published in Annals of Biomedical Engineering, this whole data set, so you can look at these. But it just makes this point that macrophages and pores in the pores show high M1, low M2, and outside we see a high M2. And 35 micron pores do have most vessels and least fibrosis. So consistent. So we have many questions about these macrophages, but you know, another set of questions. If we put it into skin, we get skin. If we put it into bone, we get bone. Put it into heart, we get a reconstruction of heart stroma. Put it into vaginal wall, we get something that looks like vaginal wall. It's pretty remarkable. What's going on there? And that's, you know, a set of questions we keep on asking is, why does it regenerate the tissue that it goes into? Where does it get its signals from to know it should make skin and not sclera or something? So anyway, kind of a mystery, and we think a lot about it. I got some hypotheses we can talk about later if you want. But in any event, for taking this to tissue engineering, since we are seemingly reconstructing tissue, we figured we needed a biodegradable form of this material. So, and this polyhema, the material of the soft contact lens, is, as they call it, remarkably biostable. Holds up very well for long periods of time. So we developed some new chemistry, which has the polyhema chains connected by degradable cross-linkers and degradable backbone units that fall apart into little units that are soluble and can be cleared by the body. And you can try to see this. Let's see if this is going to run here. It's a movie, yeah. And so what we're doing is accelerating the degradation on a stage of a confocal microscope, actually. And the bottom line is the whole thing's gone, completely gone. It completely dissolves this biodegradable form. So we did that by increasing the pH. So I'm going to, in the last 2 or 3 minutes, talk about a little bit about one other subject. It's not the only way we found to eliminate this foreign body capsule. Back in 1995, when the National Science Foundation was site visiting us for our UWeb center, we proposed a hypothesis. We said if we could make a material, we said that the protein-coated material seems to be what the macrophage is attacking. We said if we could make a material that absorbed no proteins, the body wouldn't see it, and it would just sit there in a totally inert fashion. So we started looking at this idea of non-fouling materials, and really the first paper that we found in the literature for non-fouling materials is really this one by a Japanese group. Came out in the American Chemical Society Polymer Preprints in 1983. I think it was quite a classic paper in showing how you could make a solid surface that resisted protein absorption. But there were, and this has led to kind of what I call a world of non-fouling surfaces. You can see a huge number of possibilities. Don't have the time to read them all. Many possibilities here. But we continued doing some experiments, and this is a very complicated slide. And I spent so much time talking about the slide that I decided to make a cartoon. So there's real data. Again, it's published. But the cartoon will explain what's going on in those data tables. So what we found is that you could make these non-fouling surfaces with self-assembled monolayers. We found a way to put a glow discharge plasma deposit that's self-assembling. We could make various types of brushes that are self-assembling. And we examined these under, most research on non-fouling surfaces is done with dilute protein solutions. The reason why is concentrated protein solutions cost much, much more. And also, they get very viscous. It makes it hard to work with. So people have always used maybe 1% plasma, things like that, for these types of experiments. And if you use 1% plasma, you find these head groups on the SAMs resist protein pickup. The plasma deposits resist protein pickup. The brushes, they all work fine. If you go to full plasma concentration, though, the brushes totally fail. Our plasma deposited films totally fail. But did I say brushes? I mean the self-assembled monolayer. But the brushes do continue to function. We know a lot about this, but the bottom line is these are the only types of materials, really, that will function in the body for long periods of time at full, obviously we have full plasma concentration without fouling. And so we did some experiments here. It says submitted, now it's since been accepted. I should have updated this slide. We used the polyhema material, the hydrogel, with a PEG crosslinker, polyethylene glycol crosslinker. And then my colleague, Xiaoyu Zhang, developed this very, very clever material. It's called sphincterionic material. It has plus and minus charges in equal numbers. It's actually very similar to glycine, a glycine-like structure. But anyway, we implanted these two materials, the polyhema, a classic hydrogel, and this new type of sphincterionic material in a brush form that resists all proteins. And these were 3-month implantations. And the polyhema, the hydrogel is here, dense foreign body reaction, and then you can see some muscle tissue and such underneath. But here's the foreign body capsule. And then we implant the sphincterionic hydrogel, and you see there's no capsule. So if we look at collagen density as a distance from the implant, it's very high on the polyhema. It falls off with distance as we go away from the implant. Where this carboxybetane material shows almost no capsule. And we could see this, this is blood vessels, and the carboxybetane has a lot of nice blood vessels in the vicinity of the implant, where the polyhema has a very few. And we looked at the M1, M2 macrophage. And sure enough, the colors are weird, but these 4 bars, 1, 2, 3, 4, are all carboxybetane, and the other bars are polyhema. And look at all the M2 here with the carboxybetane, a few here. And again, M1, the polyhema has the M1, much fewer M1 for the carboxybetane. And then we've got this mixed phenotype we're still trying to figure out. So let's see. I have reached 57 minutes. That's enough time. I'm going to skip this next subject. I got just a few slides on some work we've done using biological signaling to also do the same kind of work, and said we might be able to send a signal, a biological signal, and get these results. And we explored a lot of different parameter space, and eventually settled on this class of proteins called matricellular proteins, spark, osteopontin, thrombospondin. We're all interested in. And we showed that by changing, using knockout mice, we could alter the types of foreign body reactions we got. And we've published a lot of this work, too. So anyway, let me pull into the home stretch here. An inert synthetic polymer can have almost a pharmacologic effect in controlled healing and regeneration. Scar healing can be replaced by vascularized, integrated healing. There may be multiple ways to achieve this goal. We've certainly shown a few of them. Cells, a lot of people do tissue engineering and put in cells. we're wondering if all we maybe need is the macrophages. And the importance of M1, M2, I think needs some clarification. This is about half of our biomaterials group at the University of Washington. It's not my group, it's many professors here. This is Alan Hoffman, who really founded the entire group in 1970. We have quite an effort going on here. This acknowledges a nice picture of Mount Rainier, but also acknowledges our funding agencies, the National Science Foundation, the NIH. And finally, let me just put up an ad for a biomaterials short course that we teach every summer. Seattle's not so far away. If anybody would like to come, we have some very good prices, and we call it an intensive short course because for 2.5 days we work you pretty hard, but you get a good feeling for the field. Okay, well, thank you very much for your attention. And if there are questions, I'd be pleased to take them. Yeah.
The M1 and M2 macrophages, do you know if they are different cells that arrive or if one turns into the other one?
We're almost certain the M1 transitions to the M2. The M1 is critical to normal wound healing. And what we think happens in normal biology is when you get normal, uncomplicated wound healing, first the M1 comes, attacks the bacteria, cleans up necrotic tissue, and then transitions to the M2 to induce the healing and regeneration, normal wound healing. Yep. Jill.
That's fantastic. All the different classes of macrophages that are in just about every tissue, M1 and M2 are in all those different macrophages?
It's an interesting question. They've certainly been noted in peritoneal macrophage populations, alveolar and the soft tissue macrophage. I don't know if, we have cells we call histiocytes and osteoclasts are another class. We don't know if they may. We just don't know.
I think I might look into it.
Yeah, would be certainly interesting.
And I have another question, if no one else is greedy. The hydrogels that you're using, you use those around your spheres too, and then dissolve the spheres.
That's
so
right.
You actually have a hydrogel there.
Yeah, it's really a hydrogel sponge, if you want. It's just a very precision sponge, since we have this control of pore sizes. It feels like a sponge.
Lovely.
Yeah.
Thank you.
Okay, many thanks, Matty, for a fascinating
Well, good.
and Matty'll be around for a little while if anyone's looking at this.
You've got to eat my chocolate cookies.